Hello viewers, in this particular lecture on bacterial reproduction, I shall be discussing some aspects of transduction and then the aspects of lytic and lysogenic cycle. At the same time, I shall also discuss some aspects of sexual reproduction in archibacteria, mainly in terms of transformation. The phenomenon of transduction was discovered by Lederberg and Zender in 1952, and this phenomenon means the transmission of bacterial gene from one particular bacteria to another bacteria via a phage virus. So you can see in this particular diagram, this is a bacteriophage which is attacking a bacterial cell, and the phage DNA is entering into the bacterial cell, and of course the enzymes are degrading the host DNA, and then it is producing the phage particle, and ultimately this bacterial DNA is entering into the phage particle and is transmitted to another bacteria. So in what way we can see in this reproduction occurs that a particular bacterial gene is gradually getting transmitted from one bacteria to other, where the medium is the phage particle. Now the phenomenon of transduction can be of two types. They are generalized transduction and specialized transduction. Now the generalized transduction is a phenomenon in which the complete bacterial gene is actually getting into the bacteriophage. You can see here this light blue color represent the phage DNA, whereas the dark blue is representing the bacterial DNA. So completely this particular bacterial DNA is entering into the phage particle and is getting into another bacteria where it is getting replicated. And this phenomenon, what is the most important thing in generalized transduction, that the DNA can be absorbed by the DNA of the second bacteria, or it can be recircularized to produce the plasmid again, or sometimes it is matching with the homologous chromosome and may perpetuate in conjugation. So there are the different fates of this particular DNA that enters into the new bacteria via generalized transduction. But coming to the next important aspects, which is the specialized transduction. As you can see, it was denoted by Esther Lederberg in 1952. And this phenomenon is something different. Here what happens is a particular bacterial genome is actually getting mixed with the phage genome and getting into the bacteria. So it is not that the entire bacterial gene is getting packaged into the phage particle and going into another bacteria, but only some specialized gene which is in vicinity to that phage genome may get into the new bacteria and perpetuate there. And this phenomenon of specialized transduction, in that way, the cell which is getting modified by the DNA of the bacteria and the phage gene, they are called the heterogenote. So this unique phenomenon of specialized transduction is definitely standing far ahead of generalized transduction, because it means transmission of some specialized gene.
Sir, I have a question.
Yes.
In transduction, does the DNase enzyme has any activity on the phage DNA?
You see, DNase enzyme definitely will cleave DNA. So when it is a naked DNA entering into the bacterial cell, that is for transformation, the DNase enzyme will be active, so sometimes transformation will fail. But here there is the bacteriophage, so automatically there is some protection, so DNase enzyme may not have the same action as it is there in case of a naked DNA. Is it clear? So now we come to the next important aspect, which is the different applications of transduction. Now definitely I am telling that a set of bacterial gene is transmitted from one bacteria to other. So thereby they will develop new properties. So what we can say? We will say new bacteria will evolve with new set of characters. There will be some metabolic modification. And in the same way, they can develop resistance to antibiotic. And in the same way, the phage particle which is invading the bacteria may also be modified. So there will be new characters which will emerge in the phage. And lastly and not the least, they will ultimately result in cure to some genetic problem in that bacteria, or in other words, it is one way towards evolution. So this is transduction. Coming to the next important aspect, which is the lytic cycle or the lysogenic cycle. But basically you must understand lytic or lysogeny, they are all bacteriophage replication. But because it is involving the bacterial cell, they are considered as bacterial reproduction or replication. Now in lytic cycle, basically there is a bacteriophage which is attacking a bacterial cell, and thereby it will undergo lysis of that particular bacterial cell, and the bacterial nucleic acid will contribute towards the synthesis of the viral genome, and ultimately the completed virus particle will be eliminated from the bacterial cell. This process is called lytic cycle because the cell is undergoing lysis. But in case of lysogeny, what happens is the bacterial cell will be affected by the virus, but that viral genome will enter into that bacterial DNA and will replicate along with it. And this condition is called a prophage. So thereby, for a temporary period, the genetic load of that bacterial cell will increase because the incorporation of that particular phage DNA. And thereby we say that this phenomenon of lytic to lysogenic conversion is very common. And say for example, sometimes lytic to lysogenic conversion may be induced by UV light or vice versa. That is lysogeny to lytic. And it so happens that, say for example, a bacterial cell will receive the phage DNA, will replicate along with it. A time will come, it will replicate first generation, second generation, third generation. Then after n number of generation, again it can revert back to lysis. That means for that particular generation, that bacterial cell will be degraded and the bacteriophage particles will come out with the degradation of the cell. Lytic cycle include phases of adsorption, meaning at a specific site in the bacterial cell, the phage particle will settle down. And then comes penetration, where the phage DNA will enter into the bacterial cell through some definite enzymatic action like lysozyme. This is penetration. And then there is a unique phenomenon. That means that particular bacterial cell will be totally submitting to the bacteriophage. And this phenomenon is actually, we call it eclipse phase, or we call it modification phase or synthetic phase, whatever may be the case. But there are a series of gene expression. And these gene expression you can see in this particular operon, there will be the E, the L, meaning there will be the early expression, late expressing gene. Early expressing gene can be immediate early gene. Immediate early gene is followed by late early gene and the late gene. So you can understand when a particular phage DNA is entering into the bacterial cell, immediately it will produce some protein factor, and this protein factor can be N factor or CRO factor, whatever. These early factors will actually progress to allow the lytic cycle to take on. Then what happens? There will be the late expression, and this late expression can be the expression of the Q gene. So N gene to Q gene is immediate early to late early. And then we come to the expression of the late phase gene. Now this late phase gene can be like this. There may be two ways. One is the production of the lytic repressor. If the lytic repressor is expressed, then naturally the lytic cycle will be repressed and the lysogeny will take over. This is one probability. And in the other probability, when this late gene they are expressing, they are producing specific protein of the viral coat. And that viral coat include the head and also the tail. So naturally these proteins will be produced, and that means I have all the raw materials inside the bacterial cell which can produce a fresh virus particle. So I go back to this slide, we can see that the phage DNA and the phage protein are produced. So naturally the next phase becomes assembly. The DNA and the protein will assemble with each other and the complete phage particle will be produced inside the particular bacterial cell. And naturally the last phase is release. In the release, what happens is the bacterial cell will undergo rupture and the phage particle containing that particular bacterial DNA modified to phage DNA will be coming out. This complete cycle or the lytic cycle may take anything between 15 to 60 minutes, and it occurs specifically at a temperature which is suitable for bacteria, that is 37 degrees centigrade. Even the temperature is same as that our human body is having, 37 degrees centigrade. So bacteriophage can be also active in human body. We will come to that phenomenon later on. But what is important, that a particular bacterial cell can give rise to 250 to 300 phage particles within a span of 15 to 60 minutes. So lytic cycle is a unique phenomenon by which the bacterial cell is undergoing lysis and the phage particles are released from the complete bacterial cell. You see, when a particular bacterial cell is attacked by the bacteriophage, it is not that bacteriophage will be able to penetrate. Sometimes there may be some resistance. Now there is a unique case where a bacterial cell is offering resistance to the initiation of the lytic cycle. And this particular case is observed in a bacteria called Streptococcus thermophilus. Of course, it is a thermophilic bacteria where we have a unique sequence which is called cluster regularly interspersed short palindromic sequence, or in short it is called CRISPR. Now it is observed, you can see in this particular diagram that this is a normal infection. That is, the phage is sitting on the bacterial membrane and it is entering, and the normal phage particle will be produced with the initiation of the early transcription. But in some cases where there is spacer acquisition or there is the case of protospacer acquisition, there will be the normal transcription will not take place. So these particular cases where the bacterial cell or the cell membrane is having the CRISPR sequence is not allowing the phage to enter. Similarly, if it can be so that the protospacer was present but may be lost or may be mutated, then again there will be normal expression of that particular bacteriophage in that bacterial cell. So thereby CRISPR is a unique phenomenon which can be a resistance towards the induction of the lytic cycle.
Sir, I have a question. How does the CRISPR sequence induce resistance into the host cell?
You see that CRISPR sequence, I told you, it is the sequence of spacer or protospacer. So whenever it is acquisition of that particular bacterial cell from that phage, so if that protospacer or spacer is actually acquired by the bacterial cell, then it will not allow the bacteriophage to enter. So definitely we can say this acquisition occurs when there is actual physical contact brought about during adsorption of the phage to the bacterial cell. Clear? Now coming to the next important phase, which is the lysogenic cycle, which is another important event in the replication of bacteriophage, or in other words, in the bacterial replication or bacterial reproduction. Now lysogeny or the lysogenic cycle is exhibited by this particular lambda phage. So lambda phage is showing this cycle. What happens is the bacteriophage is attaching to the bacterial cell, and then the DNA will enter. That means there will be adsorption and penetration, and then there will be circularization and then integration of the phage DNA into the bacterial DNA. And this particular phage DNA will now undergo replication. So what happens is there is a unique phenomenon by which there will be no lysis, but the cell continues to divide. That is, the bacterial cell continues to divide along with the virogene or the phage gene. And it so happens that there may be an important event, we call it lytic to lysogeny or lysogeny to lytic cycle. That may be interconvertible stages. What happens is, say for example, if it is expression of the CL gene, that CL gene will have the lambda repressor and they will actually bind with the OR2 or OR1 promoter sequence, as a result of which the lambda repressor will be expressed and there will be no lysis, there will be only lysogeny. And then if there is expression of the CRO which will bind with the OR3, then naturally this particular repressor cannot act, so there will be lysis. Or in other words, the lytic repressor cannot act, so the lysis will be initiated. So we get into that particular lytic cycle where the cell is undergoing lysis. So we can see whenever a particular bacterial cell is attacked by the bacteriophage, it can undergo lytic cycle, undergo lysogenic cycle. But if it is undergoing lysogenic cycle, then there is naturally the expression of the cell, and whenever there is expression of the CRO, it will get back into the lytic cycle. So this interconversion is common. Now we come to the next important phenomenon when the DNA is damaged. You see, the DNA sometimes may be damaged. Now what is happening? If the DNA is damaged, then there will be CL repression, and if the CL repression is there, there will be automatically CRO activation. Now you must understand why this particular CRO activation takes place. You see, if a particular DNA of a bacterial cell is getting damaged, then the damaged DNA will continue to perpetuate. So obviously the cell will prefer that the cell will be digested and lost, and that's why the lytic cycle will set in and the lysogeny will be repressed. So this is definitely an important phenomenon where the damage in the DNA in the bacterial cell will set in the lytic cycle. There may be different application of this lysogenic cycle. What happens is, I told you that there will be a particular foreign DNA which will enter into the bacterial genome and will continue to replicate. So there must be some modification noted due to that entry of that foreign DNA. One such modification is observed in Corynebacterium diphtheri, this bacteria, where if the beta phage is actually attacking this bacteria, then they will become more pathogenic. Rather, they will start producing this diphtheria toxin. You can see this is the different parts of a diphtheria toxin, which will be produced by that particular Corynebacterium under the action of beta phage. So those bacteria which are attacked by beta phage will be more pathogenic than others. Now that means that this is due to the phenomenon where the virogene, you can see here the viral DNA is entering and this penetrating the bacterial cell or the host cell and is getting modified. So when it is entering into the host cell, it can be higher animal cell. So this type of lysogeny in the higher animal cell DNA will modify it, will modify the rate of replication, as a result of which there may be cancer induction. So this is a very important phenomenon where there will be cancer induction. And another important aspect is the production of the Clostridium botulinum toxin. Now what is happening is, this is the botulin toxin. It will be seen that those which are lysogenized will produce more botulinum toxin than normal cell. And coming to this Streptococcus infection, that is normally Streptococcus is producing a toxin which is called Streptococcus pyrogenic endotoxin, or is commonly called SPE. Now this is the SPE toxin. The production of this SPE toxin increases because of this lysogenic property of that particular bacteriophage on Streptococcus. Or in other words, the infectivity of Streptococcus increases due to the lysogenic action of that particular phage particle in that bacteria. There are also positive side. Now you can see this is particular, some character which is not present in the bacteria will be coming in. You can see that some bacteria can utilize galactose while others cannot. So what is happening due to specialized transduction, what happens is that particular phage particle will carry the gal gene, and this gal gene will enter into the new bacteria which originally cannot utilize galactose. But because of the incorporation of the galactose gene, they will now be able to utilize galactose. So you can see that it is not that bacteria is only damaging, but the bacteria is also gaining by lysogeny, and thereby it will acquire new properties from another bacteria. So these are the different aspects of lysogeny. Now a very important phenomenon comes into my mind, which is the actual utilization of phage. So you can see in this figure, you can see a bacterial cell is actually being surrounded by numerous millions of these bacteriophage particles. So naturally this bacteria can be killed. So this idea was used in the phage therapy, because particularly when this era is of antibiotic resistance, there must be some useful implication of phage therapy. But there are its own advantage and disadvantage. You can see here I have enlisted it. It can be very specific. It can replicate at site of infection. It is natural, it is safe, and of course it is useful in antibiotic resistant bacteria. These are the advantages. But there are also some disadvantages of phage therapy. And what is that? The disadvantage is that it is still in a very nascent state. There are still adequate scope of research, and at the same time there will be some toxin generated from the lysis or lysed bacterial cell, and it is normally not accessible to intracellular pathogen. And at the same time it is sometimes very difficult. Practicality of phage therapy is not yet been discussed. But maybe, necessity is the mother of invention. Maybe in future phage therapy will be replacing antibiotic therapy. It is really a wonderful thing to know that it is not only the bacteria, but archibacteria also can exhibit transformation. And there are several plasmids like PING, like the PKEF, like the PHV, which are functional plasmid within archibacteria. One such bacteria is the thermophilus. This thermophilus archibacteria will have a specific circular plasmid having several subsections. One such section is the main component of conjugation. So that means they might show conjugation. One such section will initiate the origin of replication or the ORI sequence. And one such section will be actually involved in replication or producing protein required in replication. So this property can actually be transferred from one bacteria to other, rather one archibacteria to the other. And they are actually understood in the form of short, regularly spaced repeat. So this particular short, regularly spaced repeat is, you can see here, these are also present in bacteria, but more common in archibacteria, or rather thermophilic archibacteria. Like it is also found in sulfolobus, where there is a definite conjugation event noted. Now this particular event of hexameric repeat actually indicate that this particular repeat will make the plasmid in good condition and thereby will help in conjugation. This is of course the sulfolobus plasmid. You can see this is the circular plasmid with the forward and the reverse gene and the TRA or the TRA-G or the TAB-E segment. The different areas of the plasmid which is isolated from the sulfolobus. So which indicate that there is conjugation in this particular sulfolobus bacteria. But the most important bacteria, which is an archaea of course, is the halophilic ones. Why halophilic? Because they are capable of surviving in high saline condition. One such bacteria is Halophilus salinarium, where there is a unique gene called VAC gene. And this VAC gene can be transmitted, or the PVAC or the CVAC, or sometimes the MCVAC, which is present in Halopherax. They can be transmitted into a recipient Archibacteria. Here it is halophilic or Halophilus volcani. And this transmission definitely is through a 150 kb plasmid, PHH1. And whenever this particular plasmid is transmitted, what is going? Sometimes the tolerance to saline condition may be acquired by the recipient.
So how do we know that the VAC gene has actually been transferred from one archaea to another?
Yeah, that's a good question. You see, whenever I told you VAC, the name comes from vacuole. Though it is allowing tolerance to saline condition, but the VAC gene will actually induce the production of the particular pseudovacuole or the gas vacuole. So if the PVAC is transmitted, the vacuole shape will be spindle shaped. If the CVAC is transmitted, the vacuole shape will be cylindrical. So by seeing the shape of the gas vacuole, we can say that this particular transmission is successful from one archaea to the other. Concludingly, what we can say that we have learned the different sexual reproductive method in bacteria in terms of transduction, in terms of lytic and lysogenic cycle, and we have also learned what are the actual benefits from these processes in bacteria and how human can utilize this particular property. And at the same time, we have concluded with archibacteria. We have shown how this archibacteria has shown typical transformation by adopting external plasmid from other donor bacteria. Okay?